I just want to talk a little bit about sort of the stuff we're doing and that is largely building on large-scale discovery projects that we've done as part of our own work and part of the Cancer Genome Atlas and things like that over the last few years. So I'll just touch on 3 different areas that to me are most exciting. Hopefully they're exciting to you as well. First is looking at breast cancer at markers for late relapse ER-positive breast cancer. Second is really in the heme malignancies. Where are we going with studying patients that have AML, acute myeloid leukemia, which is a essentially fatal disease in adults. And then lastly, like turning the whole cancer genomics paradigm a bit on its head. So normally we think about sequence a tumor, identify a targeted therapy because of a gene mutation, give the patient a targeted therapy. What I'll illustrate for you now is that we're actually using the genomics still, but designing personalized vaccines for patients from the data that we get out of genomic analysis. So late relapse ER-positive breast cancer is a real conundrum. So typically women get diagnosed with this disease somewhere around 50s or 60s. ER-positive disease is largely postmenopausal. And then they may go through some surgical intervention, some reduction of estrogen synthesis using an aromatase inhibitor, for example. And many of these women do fine, but a significant number actually relapse 15 to 20 years after their initial diagnosis and treatment. So we know that one of the reasons for this is because of long-term estrogen suppression. The cancer seems to sort of invent around that evolutionary pressure, deprivation of estrogen, and mutations occur in the estrogen receptor gene that can be identified by sequencing. So these are point mutations typically in the DNA binding domain. We've also identified translocations into the ESR1 gene that have, essentially are gain of function or turn the gene on perpetually and the protein by extension. So that's one form of resistance, but it's not the only form. And so to really get at the essence of this problem, you have to have very old age samples. Hopefully they've all been treated in the exact same way, and you have to have long-term follow-up so that you know the outcome. Did the patient develop a second breast cancer or metastatic recurrent disease, or did they not? And so by inference, then you have to use samples that are very old, that have been banked a long time ago. So one example of what we've done so far here is a study from British Columbia. Torsten Nielsen is the pathologist who put together the study and then collected all of the samples, did the downstream follow-up, et cetera. And all of these women had ER-positive disease and 5 years of tamoxifen, which is an estrogen suppression monotherapy. And so we can do things, as I'll show you in a minute, that illustrate the connection between specific mutations and long-term outcome. And so then the question is, what do you sequence? So one thing you could sequence would be the whole exome. But we felt like when we started this project a couple of years ago, a year and a half ago, that there was so much known about breast cancers from over 3,000 samples that have been sequenced, most in the treatment-naive setting, that we could probably coalesce around a much smaller set of genes that would be highly informative. So we kind of put our thinking caps on, and this is not just us, but this is in collaboration with people like Torsten, Matt Ellis, who's a breast oncologist that we've worked with for a very long time, et cetera. And we essentially looked through all of the significantly mutated genes in breast cancers and came up with a set of 83 that include things like ESR1, for example, as you'll see. And so we had those synthesized as a probe set from IDT, and off we went. So I'll show you some of the results from this. So this is a challenging approach technically as well, because as you can imagine, breast cancer samples that are 10, 15, 25 years old are fairly degraded. They've all been put into formalin blocks. They've all been stored for long periods of time. And since they were collected so long ago, nobody was doing sequencing at the time and knew how important it was to have a matched normal sample. So none of these women provided a blood or saliva normal sample as part of the clinical trial. So we basically don't have a comparator normal at all. So we start out with a large number of samples, and we evaluated the quality of the DNA coming out of those and the amount, and we were able to put these all through an automated library construction process on a robot, which you would have to do for 600 plus samples, or your technicians would quit. And so we were successful at making across a range of inputs. We pooled these together as barcoded libraries, 96 samples together at a time, and then we captured them with this 83 gene panel from IDT. And so here's some of the metrics of the data analysis along the lines of what we were just talking about a few minutes ago, depth and breadth. So 80% of bases covered at least 20-fold depth is sort of our metric for success here from any capture type of sequencing reaction. As you can see, we came out with a very high percentage of samples, 99%, that actually passed our QC metric, which is here. The number here, 632, is different than the starting number just because we had some fallout for samples that didn't meet our quality criteria for the DNA going into the libraries. Yeah?
So how do you determine what counts as passing quality control? And how do you come up with like 80% of bases?
So you align the reads, and then we just have a large-scale algorithm that looks at breadth and depth and calculates that for every sample, and then puts that out. It's called RefCov. I mentioned it the other day. And it puts it out as sort of a color-coded chart that the production manager can just look at and say, yes, all of these samples pass, or some proportion thereof. It's pretty straightforward. Yeah?
And so these are from their original, the FFPE from their original tumor.
Oh, yeah. Yeah. So these are all primary tumors. They're all treatment naive. We just know the long-term outcome. Yeah, it's a great point to emphasize. The other thing is that, just a second, the other thing to emphasize is just that even though we had fairly low inputs for these libraries, the duplication rate turned out to be extraordinarily low. So this would be like your best case scenario for a whole genome library. And whenever you go through, capture, you sort of can add up on the duplication rate and that sort of thing and be less unique, but the duplication rate overall was quite low. Thomas?
Do you know the average coverage rate for these samples? Or how deep do you usually sequence?
Yes. So these ended up being on the order of about 150-fold depth on average. Yeah.
Do you think this is sufficient, or would you increase it today? Like the first exomes were all 30x, and then we realized it didn't work. Then it's 70x, now it's 150x.
Yeah.
So, and the people at Sloan Kettering, they're always sequencing
500x, yeah. So it's a good point. I think given the lower input, we would probably always like to sequence to higher depth, but I think the 100+ fold is enough to give you, maybe down to 10% variant allele fraction capability. So you can't detect super rare subclonal mutations, but you can detect reasonably, 10% of the cell population contributing a rare variant. So I think it's a great question, and I would always love to have more coverage, but sometimes you just can't get it.
150 input.
Oh, yeah. Okay, so I'm not going to go through this extensively, but just to kind of point out a couple of things in terms of how much extra work we had to go through to get a reasonable feeling for somatic versus germline mutations. Because remember, we don't have the germline match for these patients, so how do you get the things that are frequent in the population actually removed out, so you're not reporting things that are inherited as opposed to tumor unique. And so this is kind of the overall flow cell of the experimental design, talking about selecting the genes, and then I'll show you data from our survival analysis and mutation analyses. But I've kind of broken out this filtering step just to give you an idea of all the different things that we go through. So there are lots of large-scale projects now in the publicly available data sets like 1,000 genomes that will allow you to correct for relatively low minor allele frequency germline variants, okay? So here we use a combination of the 1,000 Genomes Project, which was at that time 1,092 in the reference data set, and then NHLBI exomes, which were obtained largely from the Framingham and other cohorts that have been collected by Heart, Lung, and Blood over the years, just sequenced. And these are people who have things like, you know, hypertension, but they don't necessarily have cancer. It's still not a perfect data set because, of course, we don't know whether any of these people went on to develop cancer. But to a first approximation, we don't care because we're really just interested in subtracting out germline variants, pure and simple. We also wanted to look at artifacts that were unique to breast cancer data sets in terms of normal genomes that did get sequences match comparators. And so that came from TCGA. So we have lots and lots of samples sequenced by exome in TCGA, fewer samples by whole genome, but we also included those in just because sometimes with whole genome data you can pick up things that you can't pick up with exome sequencing. And so those are used to remove artifacts. The next thing we did is kind of unique, I think, so I'll talk a little bit more about that. And that's what we call knowledge-based variant detection. So as you guys are going to learn about over the next few days, whenever you go to detect variants, let's say in data from exome sequencing, you have to set thresholds for what you believe. So how many reads do I need to see through my algorithm that's examining the data that have a variant allele represented or a mutation, whatever you want to call it, before I believe that that's actually real as opposed to an artifact, just some substitution mutations that got, you know, crept in the source, in the course of doing sequencing, et cetera. So whenever you set these thresholds, you have to accept that in some cases, for example, you won't reach that threshold, but you'll actually have a real variant in your data set, and it'll get missed, okay? So that's a false negative result, which is just a consequence of setting thresholds at an arbitrary level. And you have to do that because the exome's too big to go look through everything with IGV across 600 samples, for example. You just couldn't do it. And so that's what you have to do. So that's, though, a bit dissatisfying because if a mutation is there, you want to see it, even in the research setting, but especially in the clinical setting, right? I mean, errors of false negativity, so missing something are sort of a really bad thing. So what we did is we came up with a knowledge base of different mutations that are identified frequently in cancers that are prognostic or diagnostic or have some functional relevance. And this is a database called the Database of Curated Mutations. This is a link to it here. And it's always moving on, so these numbers are inevitably out of date. But at the time I put this slide together, my graduate student did, we had 751 variants and 74 genes curated across 42 cancer types. So if you want to ask me why we don't just use COSMIC, I'm happy to tell you. COSMIC is kind of a cosmic waste dump, so everything goes in there. These are highly curated variants that have something to do with a therapy, prognosis or diagnosis in the disease, or some functional evidence from like cell lines or mouse models that they're actually relevant to cancer. And therefore, you would not want to miss them if you were looking for cancer-specific mutations. And in particular, what we can do with DOCM is it's not only available as like a database that you can go look at mutations in, but it also is packaged up as what we call an API, an automated programming interface. And what that means is we can put it at the end of our pipeline, and it can go back and examine all 751 variants to make sure that we didn't miss anything just because of the artificial thresholding. Okay, so it'll go back and look for evidence, and it'll point us to things that actually look like they're real. They just fell below our thresholds. So it allows us to kind of rescue mutations, if you will, back into our data set that wouldn't be there otherwise.
And this database is cancer specific?
It is. Absolutely. Okay, so we did all of that stuff, and we came up with a list of mutations across all our 600 plus samples, and then the question is, well, how do we do? And so one of the things that you can compare back to, which is what's shown here, is we compared it back to the gene list from TCGA breast cancer. Now, to be completely clear, we're looking at ER-positive disease only in our study, whereas TCGA was across all different subtypes of human breast cancer, so including triple negative. So there will be some differences, but largely we recapitulate the gene list in terms of significantly mutated genes from TCGA, where PIK3CA and MAP3K1 were the two highest. MLL3 is inserted in here because it's more specific to ER-positive disease, for example, than to all types. There are some other interesting ones here. I won't spend a lot of time on them, but MALAT1, for example, is a well-known long non-coding RNA. So it was actually very frequently mutated in these samples, et cetera. So this is kind of one way of quality checking what you got for the bulk, you know, of all of the samples. Yeah. So each one of these tick marks represents a person. Yeah, a patient. So that's why you sort of start up here with the prevalent mutations, and then they kind of get more sparse as you go further down the chart. So what did we find? Well, a few surprises, actually. So one of the things that we found is, you remember I mentioned ER, the estrogen receptor, and you know, if you look at TCGA, there are a few spurious hits to ESR1, but not many. And it's certainly less than 1% mutated in TCGA. We found that actually about 2% of samples indicated ESR1 mutations, most of which look like they're subclonal. So they're in a small minority of the cells that are contributing to the sample. And some of these, the ones in red, have been reported as rendering the gene and the protein hormone resistant versus continuing to be hormone sensitive. So there's a lot known about these mutations. But you could make an argument that, for example, if this really is at 2% in primary breast cancer, these are potentially women who should be watched carefully or kept on estrogen suppression for a much shorter period of time so that you don't sort of encourage the outgrowth of these subclonal populations by removing estrogen completely. That would have to be tested in a clinical trial, but you know, 2% of a highly prevalent tumor type is actually quite a bit, so you could probably accrue patients pretty readily onto that kind of a trial. Here's another gene. You probably know it as HER2, but it's ERB2, which is in the EGFR family. And here again, we actually had the first report in the peer-reviewed literature of point mutations in HER2. This is, of course, frequently amplified in HER2-positive breast cancer and treated with HER2 agonists. The point mutations render to targeted therapies like nivolumab. No, it's not nivolumab. It's, I can't remember the name of the drug right now, but anyway. Sorry? Well, no. Herceptin is for the amplifications. There's a small molecule that's been evaluated through clinical trials for the point mutations. It'll come to me in a minute. So here, when we originally reported this, then when we went back and looked through in TCGA samples, we estimated about 2% prevalence, and in this data set, it's about 5%. So higher, again, than expected. Lots more mutations than we had detected previously, and some of which are known to be activating. So those are the ones that are in red. The others remain to be, you know, functionalized, if you will.
How many mutations do you think are somatic mutations, and how many are rare germline variants?
I think there's probably still in this data set some small proportion that are rare germline variants. It's inevitable that since we don't have the match normal, we will have some of those filter through. So we've done the best that we can to identify those.
What is your, or what do you think is the best idea to figure out which of these mutations are really disease relevant? Do we have to test all these mutations in functional assays? Mm-hmm. Or do we have to sequence just more samples that we get some hills which mutations are repetitive? Or should we do both? I
think, yeah, so it's a great question. I think the first approach is to sequence more samples. So that's what we're doing, as I'll tell you about in a minute. But I think we need more numbers behind these to begin with.
So this color is related to exotic reasons?
Sorry?
The color is the yellow and then...
No, those are protein domains.
Oh, okay.
Yeah, so this is a lolly plot which shows the gene, but also the domains in the protein that are known. Like this would be, you know, phosphorylation or something like that. Usually they're annotated, but because I've munched these into a single slide, they don't show up. Okay, so then you also find things that have never been seen before. So here's a great example. This is CBFB, which is a binding factor that partners with another protein called RUNX1 in the cell. And we identified these mutations right here at the end of exon 2 that impact the splicing on the transcripts. And so those had never been seen before, so we immediately went back to this gene in the exomes of TCGA, and what we found is that they're actually there. So this is a place where having that API for DOCM could actually come in handy. But in most cases for TCGA and the samples that had these, they were well below the threshold for detection. And part of the reason why is that the exome reagent that was used just tapered off in coverage right at the end of the exon, and so you just didn't pick them up. And I'm not dogging on anybody's TCGA data but my own, because we did all the sequencing for breast cancer samples for TCGA. So just to be clear about that. The interesting thing about this guy is that there are lots of alternative possibilities for splicing that could occur as a result of that. And I actually mentioned this result recently to Carlos Caldas from CRUK. They've done similar looks at targeted gene sequencing in their Metabrick samples. And he pointed me back to one of their analysts, and they've actually seen a lot more mutations than even we have in their samples. So additional splice site mutations here on the companion exon, which is exon 3, but on the acceptor side instead, for example, seem to predominate. So there's a lot going on in this gene that has completely escaped anyone's notice, and this is, of course, a downside of exome sequencing, just to be clear. Okay, so what are some correlations now that we can do in terms of outcome? Because this is ultimately what we want to get at, right, for these different gene mutations. And so here are just a couple of selected results that were a bit surprising. So this is a DNA damage repair gene, DDR1, DDR2 is another gene that's similar to it, often inferred in cancer. And in this particular case, you can see the Kaplan-Meier curves show us that these women actually have statistically worse outcomes with these mutations in DDR1. You can see the lolly plot down here. NF1 was also interesting. So this is the neurofibromatosis gene, right? It's a great big gene, hard to sequence. And what we found here is that if you just looked at all NF1 mutations, including missense mutations, et cetera, there was no statistical difference in outcome. But if you just look now at only truncating NF1 mutations, now things become looking like statistically significant, but you can see here the numbers are very, very small for the mutated state. So only 8 patients with truncating mutations. So, you know, it's a reasonable p-value, but it's certainly not significant, and we need to do more to really find more of these mutations and show this causality relationship between mutation status and outcome. And for both of these, I just want to point out that this is breast cancer specific survival. So this is, you know, the absence of a recurrent breast cancer. Okay, just one last gene we looked at, or I picked out for you to look at the Kaplan-Meier curves, and that's FOXC1. And the reason for pointing to FOXC1 is it's related to metastasis. So there have been studies that have been shown that this is a gene related to metastasis. So if you just look overall at mutated FOXC1, you can see a reasonably high p-value and poor outcome if you have a mutation in that gene. But we also had information of more detail for these patients, including where their recurrent disease was identified. So was it in the breast, or was it in the breast, local, regional, or was it somewhere else, brain, bone, et cetera? Right? So now when you do this analysis and you break that out in terms of recurrence site, what you find is that for mutated FOXC1, there's no impact on local, regional, or recurrence in the breast, but there's a huge impact in terms of recurrence elsewhere. Okay, so metastatic potential to other than breast tissue becomes quite elevated. And again, these are relatively small numbers. They're looking significant, especially for this result, but we need more information. And then the last thing you'd want to do is look at co-occurrence of different genes in mutated versus unmutated state. Why? Because now you can start to build a story about maybe how the mutations impact the tumor biology, and you can build a better predictor potentially around these kinds of concurrent versus mutually exclusive situations. So we were only able to do this now for the top mutated genes that I showed you, MAP3K1 and PIK3CA. And, you know, again, these are still relatively small numbers, but interestingly, if both of these genes are mutated, it looks like it's less of an impact on long-term outcome and survival than if they're both wild type and other genes are mutated, maybe like P53, for example, but we don't have enough of those examples to throw into the mix. So we'll get there. And I should point out that one of the reasons for doing this type of analysis is that years ago, we took these exact same samples and worked them up from the RNA standpoint, and that's a test that turns out to be now clinically available from a company called NanoString, which is the Prosigna assay that evaluates RNA expression from breast cancer samples as a prognostic indicator. Another type of test like this is Oncotype DX, which uses far fewer genes. And while the NanoString assay, Prosigna, does a statistically better job of partitioning women into high risk of recurrence versus low risk of recurrence, there are still women who fall into the medium risk of recurrence. So one of the reasons for doing this study is to say, if we could look at RNA expression and if we could add on to that genes that we know predict for a statistically significant worse outcome in the disease, we could potentially add those into the assay. And we might then put even additional women into the high risk or low risk category and do a better job of stratifying them, which helps in making treatment decisions long term. Yeah?
So you said you had RNA from all of these samples?
Oh, yeah. Yeah.
So could you then match up things that say had like the mutation for FOXC1, but also had, you know, those same samples had increases in certain gene expression as well, and lay that on top?
Yeah, that's the idea. So you would take the signature of poor outcome and then add in additional mutations, and ideally you do it all on the same platform. Right? Yeah.
How many milligrams of tissue did you have at this point?
Oh, it was like making core, you know, biopsies from around the sort of ragged edges of the tissue. So this is not what you would want to do, because we'd already used these samples for the registration data for the FDA for Prosigna, and now we're going back to sort of get whatever scraps of tissue we can to get DNA out of. Yeah, these are all in blocks.
Did you do any, like, looking at separate places in the same block, like
No.
from the...
No, I mean, Torsten had done H&Es off of the block, so we knew where the concentration of tissue is at.
Because if you were, like, if your border or your margin was clean, could you get genomic DNA from the margin?
Sure, yeah, you could do that. I really worry there about field effects. These have been shown to be the case in breast cancer. And if you think about it, breast tissue is really crappy tissue to get anything out of because it's mostly fat. There's not much cellularity to it at all. Yeah. Okay, sorry. So just to finish up where we're at with this, you know, this has sort of been technically challenging, but looks like it's rewarding. We're getting some good results. We actually just finished sequencing through another smaller cohort, but hundreds of samples from a similar study from the UK. So Mitch Dowsett, who's at the ICR, is also our collaborator on this project. And we have now a third set of samples that are starting to come in the door. So 200 arrived the other day from another study that Torsten did in British Columbia. So at the end of the day, we plan to have on the order of like 12 or 1,300 samples sequenced through this 83 gene assay, all with long-term outcomes, all with similar treatments, at least for the individual cohorts, and we should be able to, I think, then contribute that information to a better prognostic indicator. And for some of these samples, as I just mentioned, we also have the NanoString data, and so we can now begin to do the kind of computational exercises that will build in predictors of mutational predictors of outcome along with RNA expression predictors of outcome and so on and so forth. We have a fourth set of samples that's part of a different study, but it is also being typed now by the PAM50 assay, which is what Prosigna is, and I've applied for funding from the Avon Foundation to also do this panel on those samples too. So that would be about another 500 old age samples, yeah.
So 2 questions. Are you able or planning to go to any of these patients? So by definition, all these patients have relapsed, right? So
No, no.
Oh, I thought this was 600 late.
Their outcomes are all known, but they didn't all relapse, right? So you have some who didn't and some who did.
So of the ones that relapsed, I guess my question is, if
Yeah.
you go back and sample and see if the clones that you detected a long time ago at 1% are now are a major part of their relapse?
Would that we could. I think that was not part of the clinical trial, so we couldn't. And in a lot of times in the metastatic setting, you don't see the tumors being removed. So they may be biopsied, but I think it would be really hard to go back to those.
And
Yeah.
my other question was, did you ever look, particularly for the top 3 genes where you have tons of
Yeah.
mutations, at levels? Is there a difference, a prognostic difference between a detection of a 1% PIK3CA versus a 10%?
Yeah. Yeah, that's a good question, and I think that we can do that, but we haven't because we kind of want to have the full data set in order to do that. It's a great question, especially for PIK3CA, because sometimes these mutations are subclonal in the primary setting, but we wouldn't know what they were in the, in the relapse setting, of course, but we could look at that correlation between outcome and status, mutational status. Okay, let's talk about heme malignancies for a couple of minutes. So we've done a lot of work in AML because of our collaborator Tim Lay, who's a heme oncologist. And for TCGA, we actually contributed all the samples to the study. All 200 AMLs from TCGA were patients that were banked and treated at WashU. And this is kind of just a rundown of all of this types of data that are generated as part of one of these large-scale omics projects. And this was published a couple of years ago in the New England Journal. And this is a similar, display as to what I just showed for the breast cancer data. So this is like how you coalesce all your information. I won't walk you through this extensively except to point out that there are known fusion drivers in AML, like the PMLRR alpha, which is part of acute promyelocytic leukemia. Most of these patients are actually cured of their disease. So AML is not exclusively a fatal disease. But we've really focused more of our attention discovery-wise on patients that have highly diploid chromosomes and therefore no cytogenetic indicators of whether they'll do well or poorly in the disease. The other thing that's fun to point out here is that there were some surprises that we had not anticipated in this disease or basically in any other. So this required Hanahan and Weinberg to go back and revisit their hallmarks of cancer, because who would have guessed that mutations in cohesin molecules, which we heard about the other day, that are like pull chromosome pairs apart, at cell division and things like that, would have anything to do with cancer. But it turns out they do, and were frequently mutated. And then the other gotcha was spliceosome mutations. So proteins that participate in splicing actually are highly mutated, not just in AML, but also in some other heme malignancies, as well as in some really rare subtypes of breast cancer actually have spliceosome mutations. I just refereed a paper on this the other day, et cetera. So those are 2 kind of surprises that are now like undergoing extensive functional analyses and things like that. So looking at spliceosome mutated AMLs and what their RNA splicing patterns look like and this sort of thing. So, but for our purposes, what we came out of were significantly mutated genes, only 23. So AML is a bit of a weird disease in that the average number of coding mutations in any AML patient is 12. So it's it's almost like pediatric cancer in that there are very, very few mutations in these genomes in the coding space. So only 23 genes emerged as significant. They're shown here. But for the purposes of our clinical assay that I'm going to tell you about now, we actually expanded that out to include recurrently mutated genes, or RMGs. And so these were, when you declare significance, you basically do 3 different tests of significance that are different. And if 2 of the 3 score is significant, then you're a significantly mutated gene. That's kind of how that works. We decided that for 200 samples that were kind of split across different subtypes of AML, maybe that just wasn't enough samples to really narrow down significance. And so we decided in our clinical approach to the disease, which I'll tell you about, to expand this list out to genes that were at least mutated in in 2 samples that were sequenced of the 200. So that was kind of our minimum level of, and what we did with these RMGs is we also turned them into a capture probe set, just from the IDT, like I just told you about for breast cancer, different genes, a much larger number even than we captured for breast cancer. And we did this little experiment that's shown here, which is an experiment that shows you that stoichiometry works, right? So when you have exome capture probes, and now you add in extra probes for the 260 mutated genes, you get higher coverage on those. So it's kind of like having your cake and eating it too. You get exome coverage, so anything that's new as you go through hundreds more AMLs and sequencing will crop up from exome. We haven't eliminated our ability to find new things, but we've got much enhanced capability to identify at high depth coverage on the genes that we think we know about in the context of the disease and may want to revisit RMGs being, elevated to SMGs at some point in time. Okay. So now how do we apply this clinically? Well, we want to look at patients in AML that do the worst. So this is kind of what your spectrum looks like in terms of likely outcomes in AML. Very few patients stay in remission for 1 year or longer. Even fewer stay in remission for 2 years, okay? With the exception of acute promyelocytic, like I said, but that's a fairly rare subtype in the disease. Those patients are typically cured. So what we really wanted to do was sort of focus on these guys over here. So primary refractory disease is basically described as patients who are chemo-refractory. So they get chemotherapy, but they don't get into remission at all. Okay, so they're not responsive to the chemo drugs. Early refractory patients look, for all intents and purposes, like they have responded, but they typically relapse within 6 months. And the clinical assays that look at minimal residual disease using flow cytometry are sort of the standard of care for predicting patients that are primary refractory and early refractory and therefore likely to progress or relapse. So there are some other available predictors. They're shown over here. I mentioned cytogenetics already. This is a good outcome. If you lose chromosome 5 or if you have complex cytogenetics, that's also a bad outcome indicator. And there are some mutations that are favorable and some that are unfavorable. But the problem is that we really just are imperfect in our ability to predict how these patients are going to do. And so we wanted to make that better, or at least evaluate whether we could make it better using a genomic intermediate. So this is kind of the test that we came up with that I'll show you. And it's a little bit different than what we think about in solid tissue disease, where you want to get a biopsy of a patient's tumor, extract DNA, sequence it, and maybe see if you can find a targeted therapy that they might be able to get either as part of the standard of care or clinical trial. Here what we're doing is we're comparing 2 time points. When you are diagnosed with AML, you go almost immediately onto chemotherapy. There's no sort of decision process. You just come into the hospital and you stay for the next 30 days and you get really, really bad drugs that make you very, very sick and ruin your immune system. At the end of that time period, your marrow is reestablishing itself, and so if you still have cancer, that should be detectable at this time point. And if you're cleared and you're in remission, that should also be evaluable. So that day 30 marrow is taken as part of the standard of care because it needs to be evaluated to see if that patient has achieved remission. And we can get DNA from both of those samples. In heme malignancies, since the tumor is in the blood, you take a different source of normal, and this is typically a skin punch that's taken at the hip where the marrow is taken out of the, as an aspirate. So, pain on top of pain. So this is where we get the normal DNA. So the idea for our assay was take all of these 3 sources of DNA, run them through exome capture with the RMG probes spiked in so we can get extra depth, and then do high sensitivity variant detection, day 0 versus normal, day 30 versus normal, and compare them to each other and see what's changed in the course of that 30-day time period, if anything, and that chemotherapy intervention. So we want to compare the mutations that we're able to detect, and we're going to compare the variant allele fractions, the proportion of reads that actually support that that variant is real in the cancer sample or in the marrow sample.
May I ask again
yeah.
what the coverage was in these samples?
So for the exome coverage, it's 100-fold tumor, 100-fold normal. For the RMG capture, we're typically aiming at and getting 500-fold coverage. Yeah, so now we're down sort of in the realm where 1 to 5% variant allele fraction you should be able to detect with reasonably good sensitivity. So before you go do this in the clinic, and because we always bank marrow samples from patients throughout the course of their disease at our clinics, you can actually now go and do a retrospective look of what I just described before you go and put it towards all of your patients that are coming in the door. And so that's what we did, and here are just a couple of examples of what you find. So this y-axis is the tumor variant allele fraction, so the percentage of reads that support the variant. And you can see that in this patient, when they're diagnosed, they have sort of a spectrum of variant allele fractions. So probably this is the founder clone, and it contains a FLT3 and an NPMC mutation. And here's a minor subclone with an NRAS mutation that's detectable. At day 39, when this patient gets remarrowed, none of those mutations can be detected. Do we have infinite sensitivity? No, but to the level of our sensitivity, we cannot detect those by DNA sequencing. And this perpetuates out to the most recent sample that we had for this patient, which is, they're essentially a long first remission type patient. Contrast, of course, to this poor patient over here who has a very active founder clone, probably a FLT3 internal tandem duplication that's in a minor subclone. At day 30 in the analysis, we can see that the FLT3 ITD is undetectable, but all of these other mutations are still pretty detectable at a reasonable variant allele fraction between 10 and 20%. And no surprise, this patient comes back into clinic with frank disease at day 90. And most of these mutations here can be detected over here. The RUNX1 is now sort of in a minor subclone compared to what it was over here. Sorry? You can, but in this illustration we didn't show that, but that's part of the reason for doing the exome sequencing on either side as well. Yeah, because you might pick up new mutations. And in fact, based on our studies from whole genomes of primary and relapse disease, you should detect quite a few new mutations, many of which are just going to be passengers, detritus that's an effect of cytotoxic chemotherapy. It's just the standard 7+3 regimen, which is 2 highly, different drugs that are the common standard of care. I don't remember, decitabine, and there's one other, I can't remember the name of it.
So with your RMG capture probe,
yeah.
So you said you spiked it into your exome capture.
Yeah.
Probes. So does that mean when you sequence it, you'll just get them mixed in, but with your RMG bits more with higher depth?
Yeah, they have higher depth on the RMGs.
You still get the whole exome.
Oh yeah. Oh yeah, yeah. So that's the cake and eat it too sort of scenario. So, if we then look at this patient just as an example, right? So AML, like most cancers, is a disease of old age, and so a lot of these patients are not going to be able to go through a second round of, even more aggressive chemotherapy and survive it. So if this patient, for example, then presents with an IDH1 mutation in their recurrent disease, one possibility is that there are now therapies in clinical trials for IDH1 mutations. We might actually recommend that they go on to that clinical trial based on this result, which is all being done. These are retrospective samples, but in the real assay we'd like to do would be in the CLIA setting. So that's a result that can be written into their medical record, and then they can be referred on to a clinical trial. And one of the tools that we've come up with to help in that analysis, although I know IDH1, I'm not the treating oncologist, so I might be interested in sort of seeing what else is out there, is we've put together sort of this resource called CIVIC. It sort of builds on what I just described for the DOCM resource, but it actually, we have a different idea in mind for this, which is to begin to build above and beyond the evidence statements that we've already curated from the literature, the ability to crowdsource information into this database. So the idea would be that if you're a medical oncologist and you feel like you're seeing lots of patients coming through different panel tests, for example, and with signed off mutations from the pathologist, and you're giving these patients drugs like the IDH1 agonists, you could actually contribute information in terms of the mutations that you're seeing, the responses that you're seeing as a credentialed and identifiable individual. So this is not done in secret or anonymously. We can't have that. But this was the idea for sort of building this database out, because this is all happening very, very rapidly in a short period of time. And there's lots of knowledge in the community. How do you coalesce that knowledge into something that everybody can get access to at the end of the day? So this is another sort of intermediate database that we're putting in place to help curate information about cancer. Here's just an example. You can see I presented this slide at a meeting where Nick Spies, who's now in medical school at WashU, was really one of the primary drivers behind beginning to put CIVIC together. And this is just variant and evidence statements for BRAF variants. We're looking here at V600E, which is probably the most prevalent one. And we have a variety of different disease types, supporting evidence, whether there was a drug that was actually used, and we rate this in terms of our level of confidence, rate these different sources from the peer-reviewed literature, and so on and so forth. So this is the kind of evidence that we've already got in place, and then we want people to add new variants into the spectrum. For example, we've identified in a single patient with glioma a very unique BRAF mutation, which is a 3 nucleotide insertion right after V600. So what does that mean? Is that patient going to be responsive to a RAF inhibitor? Or should they get a MEK inhibitor, right? If I've treated that patient, I've seen that mutation, I can report it, and I may actually know information about what I treated them with and what their response was or was not. So this is kind of the idea behind that. And then the other tool that we have in place is DGI-DB. So this is really meant to kind of be a clearinghouse for lots of different databases that were never meant to talk to each other, like databases about drugs, databases about gene mutations in cancer or other diseases. So this is not a cancer-specific database. And information from really unminable government databases like clinicaltrials.gov, which is a morass, and really usually doesn't list anything about drug-gene interactions at all, unless you really know how to get that information out, which is pretty tricky. So we initially reported this in Nature 2013, sort of as the marker paper, and then Alex Walker, who's a postdoc in the lab, just got accepted, and it's now out in PubMed in NAR, the database issue for NAR, nucleic acids research, the second version of DGI-DB. So just to give you an idea, here are a couple of genes that I've typed in. This is kind of light, sorry, it's JAK2 and EGFR. So I've typed those into my search window. I can set different parameters, like if I want to see only results for anti-neoplastic drugs only, then I tick that box there, and then I hit the green button, and I get a result that looks like this. So these are just the JAK2 inhibitors that I've identified. So it lists the gene, the drug number or name, and then the interaction type, if it's an inhibitor, for example, here. And then I have all these source links that are over here. And what I do with those is, this is why this is a clearinghouse. I'm not listing any information per se in my database that's DGI. What I'm giving you now is a link to the database that I know where there's information about it. So you can click here on TTD, which is a different drug database that we have included, and it'll take you to TTD and to exactly the listing for this JAK2 inhibitor. Okay. Similarly, there's some drug bank links here. We also have links into My Cancer Genome, which tells you a lot about clinical trials and drugs and things like that. So there are multiple, I don't even remember the number now, database sources that are in here, but it's like 23 or 25, and now including clinical trials. Yeah.
Is this just for cancer as well?
No. No, this is for any disease. Yeah. It's been most highly curated for cancer, but there are other drugs. Yeah. Which is why you get this anti-neoplastic tick box, because if you want to see every drug or you're not interested in cancer mutations, then you can not tick that and get those information. Okay, so back to our patients. I showed these guys already. This is the real long-term look. So for all the retrospective samples that we sequence, and this is just out in JAMA as of a few weeks ago, we can post that on the website if you're interested. But look at the difference. I mean, event-free survival longer than a year, right? Patients are all performing exactly as you would expect based on this latest marrow that we have. A couple of patients seem to be creeping up with new mutations, or old mutations. This guy in particular has a P53 mutation that we've been able to detect for a while, but it seems to be kind of holding steady. So that might even be a hematopoietic stem cell expansion with the P53 mutation. And then these patients, of course, are the ones with the event-free survival less than a year. So you could predict that just by if I hadn't even labeled it, right? What we're doing in the clinical trial now that's open for this approach and is funded is we're going to do patient sequencing like I've described for you. We're also going to compare it to a really, really difficult non-standard approach that's much more highly sensitive than the typical flow cytometry-based MRD analysis, and that's 5-color flow cytometry MRD analysis, which is significantly more sensitive than standard analyses, but we think is a hard target to hit. So the end question is really, does genomics give you any more information than 5-color flow in determining those patients that have minimal residual disease and are most likely to relapse quickly or, are not responsive at all, which is not as hard of a thing to figure out. So that's where we're at with AML. Last couple of minutes is just now turning our attention, to, like I said, to this completely different analysis of the DNA sequence data, which is what are the proteins that are resulting from mutated genes that look the most different to our immune systems and therefore might be actually good candidates to design vaccines against that would be highly patient unique, okay? So we started this work a few years ago. This is a recent paper, but there was one previously in 2012 where we just kind of developed the pipeline to analyze genomic data and predict neo-antigens, okay? And so the idea here is that we take tumor and normal DNA. This is in a mouse model of sarcoma that results from exposure to a carcinogen called methylcholanthrin. So it's got lots of mutations in it because of the carcinogen exposure. And what we did with this approach is we did tumor versus normal exome sequencing. And then here we actually started with cDNA from the tumor so we can detect what's mutated and expressed. And once we identify those, then we take them through an epitope prediction algorithm. What the epitope prediction algorithm does is it says, let's look at the mutated peptide that could be bound by these immune molecules in the MHC versus the wild type peptide, and let's see which has a differential affinity in the mutated state. So those are the most likely to bind strongly and be presented as antigens for response by the immune system. So that's all that's involved, is you put in the MHC type for the mouse, you put in the list of peptides, mutated versus wild type, and you let the algorithm go to town and it puts out a list. You filter that list to remove sources of known false positivity, which we didn't know immediately. We had to iterate on this multiple times. And then you synthesize peptides that correspond to that top list of mutated epitopes. And then you evaluate these in a variety of different types of assays. I won't bore you with the details. You can read the paper. But what we did at the end of this were in vivo analyses that took two flavors. One was if you had mice and you actually took the cell lines that are grown out from these sarcoma-like tumors in the mouse and you use the peptides that you identified. These are the two mutant peptides, LAMA4 and ALG8. And you synthesize those and you give them to the mouse as a vaccine, and then you challenge the mouse with the tumor cells. Do they survive or do they not? And the answer is they do, but not with the control vaccine. In the more germane example that you would use for a human patient, though, if the mice are growing the sarcomas, and then you use this as a therapeutic peptide vaccine, do they defeat their tumors by virtue of waking up their immune system? And the answer is yes. So that was a nice proof of principle. What about in people? So we took this into a small FDA-approved clinical trial with our collaborators in melanoma, Jerry Lynette and Beatrice Carino, and this was just published in April of this year. So what we did here is we had metastatic lesions from patients that we sequenced the same way, exome sequencing, look at tumor versus normal, identify mutations, look at RNA-seq here as a standalone to look at expressed mutations and mutations that are expressed at a reasonably high level. We don't want to be sort of targeting proteins that are fairly rare. And then we evaluate these algorithmically, just like I pointed out. So same way. We know the patient's HLA type. And actually in this approach, we confined patients onto the trial that were only a specific HLA type. Very common to Caucasians, who of course are more susceptible to melanomas anyway. But we knew that the predictor, this predictor is actually best for HLA subtype A0201. So that was a requirement to get onto the trial. And then, so coming out of this, of course, as I mentioned, we get this sort of list of, peptides that are most likely to elicit an immune response, but we also went back into specimens that were taken from the patient called apheresis specimens, and we co-cultured these with little mini gene constructs that contained the different epitopes to identify whether there was any existing ability to mount an immune response. So this is using an assay called ELISpot that evaluates for interferon gamma release. The other thing that we did is, because we know that tumors are heterogeneous, right? Different cell populations, different mutational profiles. We wanted to kind of try and evaluate from the vaccines that we identified for more than one sample from each patient. So these are 2 of our patients in the trial. The total number reported in the science paper was for 3 patients. We've now done the same approach for a total of 5 patients. And what we did for these 2 patients is we actually had multiple lesions that had been biopsied at different points in time. So we sequenced those in the way that I just told you about. We used the neo-antigen predictor for HLA-A0201, and so these are the candidates that come out of that prediction. And then we apply on top of that this metric of RNA expression of the variant. So you can see how the numbers really crunch down pretty low very, very quickly, but nothing so severely as with this filter about RNA expression and RNA level. Why is this important? Well, in melanoma, which is associated with UV damage of the DNA, you have an extraordinary mutation rate, right? I mean, these are just missense mutations, so there are many more mutations than those. We have to have some way of filtering out candidates that are simply not being expressed by the tumor. Right? And so this is the way of doing it. And now the Venn diagram here is sort of looking back, and I'll show you these data in a moment, but looking back at the neoantigens for which we did get a response. There are 3 for this patient and 3 for this patient. Kind of where did they come from, and were they shared across different tumor isolates? Where most of them here were shared, not all, but some of them here were shared, and there were sharing between different isolates that doesn't include all 3. But at the end of the day, what you really want to do is, as much as possible, identify neoantigens that are in the founder clone so that you're addressing, using the vaccine and the T-cell response that's elicited from the vaccine, you're attacking as many of the tumor cells as possible because of the shared mutational burden. Okay. So then just to finish up, this is kind of the paradigm that we use for vaccine. There are multiple vaccine platforms I'll mention in just a moment. The one we used here was dendritic cells. So I know one thing about the immune system, and that is that dendritic cells are kind of like the generals that direct T cells what to go after, okay? And the T cells are like the foot soldiers. So what we did in this case is we isolated dendritic cells from the patients. We cultured them under specific conditions, including adding in GMP quality peptides that correspond to our final list. Why do we need GMP quality peptides? Because the dendritic cells are going to carry those peptides into the patient in the form of the vaccine as an infusion and instruct the T cell memory to be hopefully resulting from that infusion of the dendritic cell vaccine. Okay? Part of the reason for using dendritic cells is because our collaborators had done a previous small trial with just known shared antigen peptides that are common to melanomas in a protein called GP100. And that was a successful trial. Nobody died. And there were actually 3 patients that had a response. So building on this, now we're adding neo-antigens that are predicted from genomics. And every patient also received a peptide to the 2 GP100 controls. Okay, so this is the carry forward from the previous trial. And we also evaluated for whether they mounted an immune response to the GP100 peptides. So just to finish up, we did a dextramer assays out of the blood of these patients, both before and after vaccination, to show which of the peptides we were getting a response to. This is one of the metrics of the clinical trial. We're not trying to cure anybody of disease here, so there's no clinical benefit involved. We have 2 missions. One is not to kill anybody and to show if there are adverse events. There were none. And to actually show whether the dendritic cell vaccine is working, i.e. do we establish any kind of T-cell memory to the specific peptides. And as you can see for the 3 patients, especially focusing on the post-vaccine, right, there are evidence for 3 of the 7 peptides actually giving us a response back in these patients. They're different peptides, of course, because this is highly patient-specific, but you can see this fraction of CD8 positive cells that's emerging in the post-vaccine setting that to a large extent was not there to begin with. The other thing that you can do, oh, sorry.
That slide
Yeah.
showing,
Yeah.
why would you choose dextramers as opposed to ELISpot in this situation?
Because we're looking in the peripheral blood. So this is just a way of sort of picking up circulating T cells. Yeah.
But could you not say take those, then sort them, and then do ELISpot on that? Or?
I'm not the immunology expert, so I don't know, but I think this was a more straightforward way of getting at it. And there's now a newer method called CITOF, which allows you to also do these types of measurements with tetramers that imitate the different human MHC molecules or HLA molecules. The other thing that we can do now with, in this setting is to look at the T-cell repertoire before and after vaccination using a kit here from a company called Adaptive Biosciences. They sort of provide you with the PCR amplicon primers. You do the amplification, do the sequencing, and send them back the data, and they give you the repertoire that, the diversity of the repertoire that's emerging before and after vaccine. So these are just specific peptide antigens that are shown for 3 different patients, where the black bars are sort of the T-cell repertoire, the number of clonotypes that are present before vaccine, and the clear bars are the ones that are present after vaccine. So as you can see in every case, and probably to the extreme in this patient with this responding peptide, we really expanded the diversity of the T-cell repertoire. And I don't really understand why this is good, but apparently expansion of diversity is a good thing. So you just have to take my word for it, I guess. Yeah, so to finish up, we reported this study. We're now just waiting for a funding decision on the next step for this approach, which would be to now combine for these patients a patient-specific dendritic cell vaccine with a checkpoint blockade inhibitor. And checkpoint blockade inhibitors are also a mechanism for waking up the immune system. So we would give the checkpoint blockade first, then give the vaccine. And in the melanoma setting, there are now 3 checkpoint blockade drugs that are approved for melanoma. So we can give that as part of the standard of care for these advanced metastatic patients. So just to finish then, where are we going with this? I already mentioned this guy here, but we're already doing other tumor types. So we just opened a triple-negative breast cancer trial. One of the reasons for going after TNBC is because there is one standard of care, which is aggressive chemotherapy. All patients get the same thing after they have their tumors removed. And so what we're doing in these women is we have 2 arms of the trial. They get an at diagnosis core biopsy that's taken to confirm that they have triple-negative disease, at which point they can be eligible to consent onto the trial. This is in the primary setting. So these are no negative patients that have triple-negative disease. They have their tumors removed. They come to us, me, for DNA and RNA analysis, much like I've just described for you. They then go through the standard of care, which is ACT chemotherapy, followed by radiation therapy, at the end of which they will receive their patient-specific vaccine. So this is a window of opportunity trial, and it's highly unique. Unlike the melanoma patients, these are not advanced metastatic patients. So, and we know that, we know the relapse rate for women with triple-negative breast cancer. It's typically, if you relapse, it's going to be within 2 to 3 years after your primary diagnosis. So we actually have a reasonable period of time where we can follow these patients that receive the vaccine and say, do they do better on average, the same, worse than patients who don't receive the vaccine, because there's a very large database for that. And so these are two different vaccine platforms, just to point out that dendritic cells aren't the only way to go. And actually, some people hate dendritic cell approaches because they've been used multiple times throughout the history of cancer care and have largely been failures. So this gets pointed out to me whenever I present this. I just thought I'd preempt that. Here we're doing sort of the same mini gene cassette. So we piece in the DNA sequences that encode the neoantigens behind a promoter, and then these are electroporated into the patients. And then the other approach is just to simply synthesize the long peptides that correspond to their neoantigens, make those as a cocktail and adjuvant, and shoot them up. So it's just like a regular vaccine. I can't believe this works, by the way, but it's actually been shown. And there are also RNA vaccines that follow the same kind of formula. So we aren't doing those here, but that's another vaccine platform that people are very keen to try. Primarily because RNA is a lot cheaper to synthesize than peptides and doesn't take as long. And so one of the things that you're up against here is sort of how long do you have actually to go through this whole series of steps that I've walked you through and have a vaccine ready for patients. So this is approved by the FDA, approved by our IRB, and now enrolling, and I think we've sequenced so far like 3 patients and predicted neoantigens, and those are in the process of being tested in those downstream assays that I pointed out to you. The last one, which we're not doing but I really want to do, and I'll just finish with this, is pediatric gliomas. So through the project that we collaborated with on with St. Jude over the last 5 years, we've sequenced like over 1,000 pediatric cancers, some proportion of which were gliomas. And almost to a first approximation, every pediatric cancer is like AML, very, very, very few mutations, right? So a priori, these patients are not the appropriate types of patients that you would consider to get a vaccine, but 95% of kids with brain cancer die of brain cancer. So it's a highly fatal disease. And we found this really interesting report from Joe Costello's lab at UCSF that looked at kids where they had initial mutational load in the orange bars from their pediatric glioma when it was first diagnosed, and then they received this drug, temozolomide. This is not the standard of care, by the way, for kids. It is for adults with glioblastoma, but some of these kids got temozolomide. They recurred in their disease, not surprisingly, and when that resected disease was studied, they actually had an extraordinarily high mutation rate, and that's because of this drug. So are we, by treating these kids with this very aggressive chemotherapeutic agent, actually setting them up to potentially be better candidates for now a third intervention, which would be a vaccine? I don't know. We've looked at all of these mutations through a neoantigen prediction pipeline. We don't have these patients in hand. We just have the data from their tumors, but we can make a reasonable argument that many of these mutations in the recurrent disease are not subclonal. They look like they're highly shared mutations, and they may be, this may be a good setting to try precision vaccine. The other challenge, of course, in the brain is that antibodies like checkpoint blockade inhibitors typically can't pass the blood-brain barrier, whereas T cells should be able to, and so that's another sort of strong argument for using a vaccine-based approach. So we'll see.
These are glioma patients here, or these are glioblastoma patients?
These are glioma, pediatric gliomas. Yeah. Yeah. Yeah, I know it's pretty frightening, isn't it?
Do the vaccines have any kind of like autoimmune toxicity, like the checkpoint inhibitors do, like colitis?
No. No, because the antigens are highly tumor specific, so you would predict, obviously, that it wouldn't, but it was nice to see that it didn't. We did, just for full disclosure, have one patient who died in the trial, but it wasn't as a result of the vaccine. He had a brain lesion that he had removed right before he received his vaccine, which is commonly done, and he actually died from complications of the brain surgery. So that's not good, but it happens. Patient number 5 is doing incredibly well. We're going to report this patient as an N of 1 study because we're generating all of this data, and it's actually the first patient for which we included an HLA-B haplotype responsive neoantigen, and now we've done these dextramer assays downstream and have showed that we're actually getting a nice response to the other peptides that correspond to A0201, but also to that B neoantigen. So there are some other interesting aspects about this patient that I won't bore you with, but I think it'll be an interesting sort of single report to add.
In the triple negative study, right,
yeah.
At what point do they get the standard therapy, and then you know, when they're in the window to progress? At what point in that window are they getting the vaccine? And the related question is,
yeah.
you know, is it better to do it super early or wait till? You know, how long does the vaccine immunogenicity last? Right? I mean, if you did it right, you can make an argument to do it early so that they never recur. But if you do it early, then the immunogenicity wanes and they recur later.
Yeah. No, it's a good question. The answer is they get it once they've finished the radiotherapy and we've established that their immune system is healthy again. So there will be some dropout from patients that just aren't, their immune systems aren't recovering from chemo and radiation, because we obviously want them to be able to use the vaccine, you know, to stimulate T cell production. And the second question was, when is the right time to give it?
Or just, is it known that these vaccines like last for years? I mean, the immunogenicity that lasts a long time.
It isn't known because we're too early into this, and really the report in Science was the first report of this type of an approach. In general, the immunotherapies writ large seem to incur more durable responses, but I think that's just a generalization at this point. So, you know, we really just don't know. We do know that we can actually, and we've done this with some of the patients from the melanoma trial, we do know that once we actually have evidence that they have a CD8 plus response, we can take specimens from those and culture those in the lab and actually sort of freeze them down. So you could imagine sort of a booster shot paradigm, right? That we are doing that for that reason because we want to ultimately explore that. But none of the three patients that were in the Science study have recurred in their disease. Even though we weren't trying to show clinical benefit, they're all alive and well today. I did like a news talk show with one of the guys who was patient number 1 a month or so ago in St. Louis. So, you know, we'll see. I mean, I think this is the big question mark in immunotherapy across the board, is sort of what do the responses look like over a course of many years. But there are certainly some IPI patients that received the drug very early on for metastatic melanoma that are still alive today, you know, 10, 11 years after they were on the clinical trial. So it can be remarkable. That's probably not going to be the case for everybody, I think, is the upshot.